Welcome to your third lecture for Chapter 5, Biological Molecules. Previously, we discussed two classes of biological molecules, carbohydrates and lipids. Today, we are moving to a third class of biological molecules, proteins. Proteins have many different structures, and each structure has a specific function. So as we look at proteins as a whole, we can see there is a very wide range of functions. Keep in mind that proteins account for more than 50% of the dry weight of a cell. So proteins make up the bulk of an organism. When you look in the mirror, you're mostly looking at proteins. Some proteins are enzymatic, in other words, they are enzymes, and these proteins are going to facilitate chemical reactions in a cell, converting substrates into different molecules called products. Other proteins are structural, and they provide support, such as collagen and elastin. Other proteins function in storage. They store amino acids, which can be used, for example, in egg white by a developing embryo, or in the major protein of milk, casein, to nourish a baby mammal. Other proteins transport substances, gases around the body, or molecules across a cell membrane into and out of a cell. Some hormones are proteins, which are important for coordinating activities in an organism. Insulin is an example of a hormone that is a protein. Other proteins are receptors. They respond to chemical stimuli. Receptors are found embedded in membranes of cells and have very specific shapes that bind to molecules. Some proteins are contractile or motor, such as those that we find in our muscles. Other proteins are defensive, and I know you can't see that on this figure, but we will look at some more examples on the next couple of slides. As mentioned previously, most enzymes are proteins, and they facilitate chemical reactions. In this illustration, we can see the enzyme is shown in purple, and it has a specific shape on its surface called the active site, where it will bind to complementary molecules called substrates. In this illustration, there's a single substrate which looks like it might be a disaccharide, a small sugar. In the course of the chemical reaction, this enzyme is breaking the bond of the disaccharide and then releasing two products which are monosaccharides. So the example shown here is an enzyme that does hydrolysis, or a hydrolytic enzyme. Enzymes do all sorts of chemical reactions. They make bonds and break bonds, they rearrange atoms, but most enzymes are in fact proteins. Other proteins function in storage. In particular, storage of amino acids, the small molecules or the building blocks of proteins. Casein is the major protein found in milk, and this is important in storing amino acids so that baby mammals can acquire them and then use them to build their own proteins in their growing bodies. Ovalbumin is a protein found in egg white. This is important to nourish a developing embryo. Developing embryos are going to require many amino acids in order to build new cells. Other proteins are defensive. They protect us against microorganisms that might cause disease. Antibodies are very important protective proteins. Antibodies can inactivate or help destroy microbes. In the picture on the left, we're looking at a virus. Viruses have spikes on their exterior that they use to dock on host cells to initiate an infection. But if you are producing antibodies, shown in purple, that stick to these spikes, these can bind to the virus and neutralize it or inactivate it so that it can no longer stick to one of your cells and cause an infection. Over on the right, we're looking at a bacterium, a much larger pathogen, In this case, antibodies might coat the bacterium and mark it for more efficient recognition and destruction by your immune system. Some proteins function in transport. So hemoglobin is a protein that transports oxygen from your lungs to all other cells of your body. Other transport proteins are embedded in the cell membrane and can move molecules into or out of a cell. So in this picture, you have to imagine that there was a cell that was sliced in half so that we could see the layers. And we're looking at this region of the cell membrane and we're zooming in on it in the illustration so that you can see the cell membrane in more detail. And this protein that crosses the cell membrane, this transport protein, it has a channel on its interior that will allow some molecules, such as glucose, to cross the membrane and enter the cell. Other transport proteins will allow molecules to leave the cell. Some hormones are proteins, and hormones are important for coordinating an organism's activities. An example is insulin. Insulin is a small hormone protein that is secreted by the pancreas after we eat a meal. When we eat, we get higher levels of sugar in our bloodstream, and this stimulates the pancreas to secrete insulin into our bloodstream. The insulin will then stimulate other cells to take up the glucose, thus resulting in normal blood sugar levels. Contractile and motor proteins are important in movement. Some of these are important for moving cilia and flagella. Actin and myosin proteins are responsible for the contraction of our muscles. Receptor proteins are found embedded in the cell membrane and protruding from the surface of the cell. Receptors are important for detecting signaling molecules that are released by something else. For example, in this picture, we have a cell on the bottom with receptors on its surface that have a specific shape that will allow them to bind to signaling molecules, and then a certain response follows. So the cell above is a nerve cell that's secreting signaling molecules, and then these receptor proteins on the next cell bind to them and respond. This is a hand-in-glove relationship. Other proteins are structural and function in support, such as the keratin protein of the hair, horns, feathers, and other skin appendages. Insects and spiders have silk proteins that make their cocoons and webs function as they do. Collagen and elastin are structural proteins that are important in our connective tissues. There are many, many examples of protein function. We'll begin by considering the building blocks that are used to make all proteins. These are called amino acids. Amino acids are the monomers, or the single units, the building blocks that are used to build all proteins. When several hundred amino acids are joined together, we form our polymer, and in this case the polymer name is a polypeptide. Every bond that holds amino acids together is a peptide bond. So a polypeptide consists of many peptide bonds between many amino acids. Although polypeptides are polymers, they are not equivalent to proteins. A polypeptide does not equal a protein. A polypeptide is a linear chain of amino acids, but it must fold into a specific three-dimensional shape to be referred to as a protein. Some proteins consist of one polypeptide that has folded into a specific conformation. Other proteins consist of two or more polypeptides that come together to form that final three-dimensional shape. More about the monomers. Amino acids, as we mentioned, are the monomers that are used to build all proteins. Amino acids are small organic molecules, so they are built by organisms and they are built around a carbon skeleton. And every amino acid has a central carbon that is attached to 3 chemical groups. All 20 types of amino acids have a carboxyl functional group or chemical group and an amino chemical group. The way in which the 20 amino acids differ is in the third chemical group, which we refer to as the R group. Let's take a look at the next slide to see what these 20 amino acids look like. And then we will return to this slide. First, let's go ahead and draw a generic amino acid. Every amino acid has a central carbon that is attached to a carboxyl group or a COOH group, and on the other side an amino group or an NH2 group. This central carbon is also attached to a hydrogen. And then the next functional group we're just going to generically call R for radical group. And the 20 amino acids that are used to build all proteins are going to differ in their R groups, which are highlighted in this figure on the right in white. So all of these highlighted white groups of atoms are the R groups. And there are 20 different R groups. The 20 amino acids shown here are generic, meaning that they are used by all different organisms. All life on this planet uses these same generic small molecules called amino acids. However, the way in which they are joined together, the linear sequence in which they are joined together, and the types that are joined together will result in polymers that are unique to a given organism. So the polypeptides and the proteins are unique, even though they're built from the same generic 20 types of amino acids. As you look at the R groups of these amino acids, notice we can arrange them into groups based on their properties. So this upper region consists of amino acids that have non-polar R groups. The electrons are distributed evenly, therefore there's no partial charges, and therefore the R groups are hydrophobic. In the middle, we have a group of amino acids with polar R groups. So there are partial positive and partial negative ends since the electrons are not shared equally across these R groups. Recall that anything that is polar will interact quite nicely with water, and we can say it is hydrophilic. So amino acids with polar R groups have hydrophilic R groups. So do amino acids that have electrically charged R groups, which actually have full charges. Either they are acidic with full negative charges, or they are basic with full positive charges. Anything with a charge or a partial charge is hydrophilic and interacts quite nicely with water, whereas nonpolar R groups are hydrophobic. Lastly, I just want to point out the difference in the way I drew an amino group and a carboxyl group, and the way these two functional groups or chemical groups are represented in the figure. Notice that I drew an amino group with an N and two H's, but the amino groups shown on the amino acids in this figure all have an NH3 and a positive charge. NH3 plus is an amino, and so is NH2. In a cell where the pH is close to neutral, close to pH 7, it is more likely to find amino groups existing how you see them in this figure on the right, with 3 hydrogens and a positive charge. But it's easier to build models of amino acids if we stick with the amino group representation that I drew. The carboxyl group I drew as a COOH, but in this figure you see them as COO minus. In a biological system where the pH is almost neutral, it's more likely to see carboxyl groups represented as shown here with a COO minus. Carboxyl groups tend to lose hydrogen ions. Therefore, they are the acidic part of an amino acid. In fact, the name amino acid is derived from the two functional groups shared by all amino acids. The amino group and the acid is from the carboxyl group. Going back to the previous slide, you saw that all amino acids have a carboxyl group and an amino group. The carboxyl group might be COOH, or it could be COO minus. The amino group could be NH2, or it could be NH3 plus, the charged forms being more likely inside of a cell. And then the 20 amino acids differ in their R groups. There are 20 different R groups. When building a protein, amino acids are linked to each other by peptide bonds, ultimately to form our polymer called a polypeptide. Every peptide bond is a covalent bond. Peptide bond is just a very specific name for the covalent bond that is linking amino acids. And like all of the covalent bonds you've learned about so far, they result from dehydration synthesis reactions. D means removal, hydration means water. So removal of water to synthesize or to build a new bond. The cellular machinery that does this, that performs dehydration synthesis to construct peptide bonds, is the ribosome. All cells have thousands of ribosomes that create these new bonds between amino acids. The result is a polypeptide, which of course can vary greatly in its length and its types of amino acids. Every polypeptide will have a unique linear sequence of amino acids, and ultimately that will determine how it folds, the shape it takes on, and ultimately the protein that is formed. Keep in mind that a protein has a very specific three-dimensional shape due to folding, and every protein consists of one or more polypeptides, our initial polymer. In this figure, we are illustrating dehydration synthesis, creating new peptide bonds. You can see on the left that a peptide bond has already formed here between two amino acids. On the left, we have the amino acid serine, and you can see what the R group looks like for the amino acid serine. And it's already been connected to a different amino acid, tyrosine, which has a different R group. Now, in the course of a dehydration synthesis reaction, we are going to add a third amino acid to the growing chain. This amino acid is cysteine, and it has yet a different R group. So a ribosome will connect the new amino acid cysteine to the growing chain. Notice that the carboxyl group of one amino acid is going to react with the amino group of the next. And highlighted in blue are the atoms that will be removed in the course of the reaction. We are going to lose two hydrogens and an oxygen. We're going to lose water. That's the dehydration part. The result is that a new covalent bond forms or is synthesized. That's the synthesis part. And we call it a peptide bond because it is between amino acids. Now we have a tripeptide, a molecule that consists of 3 amino acids linked by 2 peptide bonds. I mentioned previously that a polypeptide is not the same as a protein. A polypeptide is the initial polymer. It consists of a linear chain of amino acids joined by peptide bonds. But that polypeptide must fold into a precise three-dimensional shape that will facilitate a specific function for the cell, and then we can call it a protein. So a functional protein is going to consist of at least one polypeptide, maybe two or three or four, that have twisted, folded, and coiled up into a very specific shape. The initial sequence of amino acids in that polypeptide will determine how a protein folds and what its final 3D shape will be, and this is essential to its function. Shown below is an example of a protein. This is an enzyme called lysozyme. Two pictures of it on the left and the right. Lysozyme is an enzyme that you produce in your tears and your saliva. And lysozyme is a hydrolytic enzyme. In other words, it breaks bonds. It performs hydrolysis reactions. The way that it does that is it has a very specific region on its surface. Here it's labeled as the groove. This is a domain or an area on the surface of the enzyme that is the active site, and its specific conformation allows it to bind to bacterial cell walls and chop them up. So in order for lysozyme to do its job, it has to have a very specific three-dimensional shape in its active site. You rarely get infections in your eyes. One reason is the mechanical flushing of your lacrimal apparatus, the tears that wash your eyeball. But another reason is this enzyme. When bacteria get in your eye, the active site of lysozyme will bind to their cell walls and chop them up. Lysozyme would not function if it lost this three-dimensional shape that is so important. So how do proteins get this specific three-dimensional shape? Well, they fold in a series of steps. Initially, we have what is called the primary structure of a protein, and that is the linear sequence of amino acids that you see in that initial polypeptide. The primary structure is illustrated on the bottom left of your screen. I'm just going to illustrate primary, the shorthand. The primary structure is this linear sequence of amino acids, and in this picture, every little circle is representing one amino acid, and then you can see it's joined to the next, and so forth and so on. As amino acids start to form hydrogen bonds with other amino acids, we start to get a little bit of three-dimensional structure. And we're moving to the secondary structure of protein folding. Characteristic of this are alpha helixes, or coils, and beta pleated sheets. Think of those paper fans you used to make as a kid. Similar shape. Hydrogen bonds are key to a protein's secondary structure. And you can see here these dotted lines. These dotted lines are representing hydrogen bonding between various atoms of the polypeptide, starting to give it some three-dimensional form. As a polypeptide moves into its final three-dimensional shape, we say it has acquired a tertiary structure. So the tertiary structure is the final three-dimensional shape of one polypeptide. And all types of bonds can participate here: covalent, ionic, and hydrogen, to get this final 3D shape. All proteins will progress through these first stages of folding. All proteins will move from primary to secondary to tertiary structures. For some proteins, that's where they finish folding. One polypeptide folded up into a tertiary shape is the whole protein. But some proteins must go one more step, and that is quaternary structure. Quaternary structure results when a protein consists of multiple polypeptides. So, two or more polypeptides. Shown on the far right is an example of a quaternary protein where 2, 3, or 4 polypeptides have come together to form the final functional protein. Not all proteins have a quaternary structure, but many do.
Proteins are building blocks that help give your body structure and do work around your body. They move molecules, they make new molecules, they recycle old molecules, and this is just to name a few of the things that they do. But maybe the most interesting thing is that these protein building blocks are themselves made up of smaller building blocks called amino acids. Here you can see a string of amino acids. The different shapes that you can see represent the atoms that make up each amino acid. And here we've highlighted 12 individual amino acids. To make it simpler, each different amino acid can be represented by a single letter. Now each amino acid is shown as a colored ball, looking like beads on a string. This makes the protein's structure easier to imagine. The order of amino acids is only part of the story. Because of the different shapes of the individual amino acids, they like to fold into even more interesting three-dimensional shapes. This molecule is twisting into several different spiral or helical shapes, and then those are folding on each other. Take a look at the three-dimensional shape as we give the protein a spin. Kind of looks like logs stacked in a fireplace. Here is the one-letter amino acid code revealing the identity of each amino acid. Again, now you see the amino acids drawn to show the position of each atom. This is like looking at an atomic skeleton of each amino acid. Just like you take up more space than just your skeleton would, see how much space each atom really occupies. This is the real shape of the protein. Now we've seen an example of a protein taking shape, and several of the ways scientists visualize these tiny building blocks used throughout your body.
One example of an interesting protein is that of spider silk. The abdominal glands of the spider secrete silk fibers, or silk proteins, that contain many beta-pleated sheets. Because of the specific structure of the silk protein, the spider silk protein, we see very interesting characteristics. The radiating strands of the spider silk maintain the shape of the web, for one, but also they are very elastic. They stretch in response to forces such as strong winds, rain, and even the touch of very large insects. Spider silk is an amazingly tough protein because of its three-dimensional structure. In fact, the military is interested in spider silk, and scientists have actually genetically engineered goats to produce it in their milk. These scientists found the gene for spider silk in a spider and transferred that gene to the goat. The goat's milk can be collected and the spider silk purified from the goat's milk and then used for things like bulletproof vests. Spider silk is stronger than steel. This figure is illustrating to you the tertiary structure of some polypeptide. We're looking at how the three-dimensional shape of a single polypeptide can form. The purple ribbon is representing to you the polypeptide backbone. So the purple ribbon represents the amino acids that have been linked together by peptide bonds, the chain of amino acids. Do you remember what cellular machinery creates those peptide bonds by dehydration synthesis? It's the ribosome. Once that primary structure of the polypeptide forms, various R groups of the amino acids start interacting with one another, and those are emphasized in this figure. Up at the top, we have an R group of some amino acid up here that is hydrogen bonding with the R group of another amino acid located at a different region of the protein. That causes the polypeptide backbone to fold, maybe into a beta pleated sheet, maybe into an alpha helix. It just depends on how many hydrogen bonds form and where they're located. To the right, we're emphasizing some other amino acid and its R group, which is hydrophobic or nonpolar. And that's interacting with another nonpolar or hydrophobic amino acid R group somewhere else in the protein. Recall that the inside of a cell and the outside of a cell are both full of water. Cells live in aqueous environments. So in order for these hydrophobic interactions to occur, they have to be buried in the middle of the protein somewhere, away from the water. Down below, we have some other amino acid and its R group emphasized, and notice it's forming a new covalent bond with an R group of an amino acid located across the way. So more covalent bonds can actually form between R groups after the polypeptide is synthesized by the ribosome. And then down below, we have another amino acid with its charged R group forming an ionic bond with the opposite charge of another R group across the way. So on this slide, you can see that all the different types of bonds are going to contribute to the final three-dimensional structure of a polypeptide. We have, of course, the covalent bonds that form the initial backbone. Then we can form hydrogen bonds between R groups. We can have hydrophobic interactions between R groups. We can form new covalent bonds, such as this disulfide bridge, and we can have ionic bonding as well. As mentioned previously, some proteins exhibit a quaternary structure, where multiple polypeptides come together to form the functional protein. The first example on the left is collagen. Collagen is an important structural protein of connective tissue. Notice that collagen is formed from 3 identical polypeptides that are intertwined, and that results in a very strong protein. Over to the right, we have another example of a quaternary protein, hemoglobin. Hemoglobin consists of 4 polypeptides that come together to form one functional hemoglobin protein. There are 2 alpha chains, shown in purple. These are 2 polypeptides that are identical. And there are 2 beta chain polypeptides that are identical, for 4 polypeptides total to make up this one hemoglobin protein. And this quaternary structure of hemoglobin allows it to bind oxygen and deliver it to all the tissues and cells of your body. Continuing with the hemoglobin example, we're going to look at how even one tiny change in the amino acid sequence can alter the entire structure and function of a protein. Sickle cell disease results from such a change. One amino acid changed in the primary structure of a polypeptide, and it has dramatic effects. Sickle cell disease is an inherited blood disorder. Hemoglobin, as I mentioned previously, consists of 4 polypeptides that have come together to form a single functional protein. There are 2 alpha chains, or 2 alpha polypeptides, each of which has 141 amino acids. And there are 2 beta chains, 2 different polypeptides, each of which has 146 amino acids. So if you add that up, 2 alpha chains plus 2 beta chains, you'll see that one hemoglobin protein has 574 amino acids total per protein. In sickle cell disease, one of the beta chain amino acids out of 146 has been altered. Let's begin by looking at normal hemoglobin on the left-hand side of the screen. We're focusing in on one of the polypeptides, the beta subunit. The beta subunit polypeptide has 146 amino acids. So if we were to see the entire primary structure of the beta subunit, we would count 146 amino acids joined together. In this illustration, we're only looking at 7 of those 146. Once the primary structure forms, it begins to fold into secondary and then tertiary structures. The tertiary structure is the final three-dimensional shape for this polypeptide. And then this polypeptide and an identical beta subunit come together with two other polypeptides, the alpha subunits, to make up the quaternary structure of your normal functional hemoglobin. And this hemoglobin is packed into red blood cells. So the normal structure for a red blood cell is a flat, disc-shaped cell that is full of hemoglobin proteins and is busy picking up oxygen from the lungs and delivering it to all your cells. In sickle cell disease, one amino acid is changed, and you can see that on the top right of your screen. We get a substitution where the amino acid valine is incorporated instead of the one that should be there in the normal or wild type beta subunit. This single amino acid substitution drastically affects the way the beta polypeptide folds. And you can see that in this illustration. This mutant beta polypeptide has a different shape than the normal wild type one shown on the left. When the mutant beta subunits come together with the alpha subunits, the entire quaternary structure for hemoglobin changes dramatically. This is a different shape, and the result is that hemoglobin proteins start to stick to each other. As the hemoglobin proteins clump together inside of a red blood cell, they change its shape. So the red blood cell shape takes on this sickle shape instead of the flat, circular disc shape we need for proper functioning. Mutant hemoglobins stick together, they change the shape of the red blood cells, and there are many problems that result. For example, the mutant hemoglobins have a greatly decreased capacity to carry oxygen to the tissues. And because they are changing the shape of the red blood cell, these sickle-shaped cells tend to stick in capillaries and cause other problems. The take-home message is that the initial primary structure of a protein, or the way in which the amino acids are linked together initially, is absolutely vital to the final three-dimensional structure and thus function of every protein. So what determines the structure of a protein? How does it fold through primary, secondary, tertiary, and sometimes quaternary forms? What affects the folding process? Well, there are many things that are important. pH is one. If the pH swings too acidic or too basic, proteins will not fold properly, and therefore they will not function properly. Salt concentration is important. It affects the amount of water that is there and changes the shape of proteins. Increases in temperature will drastically affect the structure of a protein, and that's shown in the figure below. If the temperature rises too much, it will cause what is called denaturation, or unfolding of the protein. An unfolded protein or a denatured protein is not active. In some cases, a denatured protein can be renatured or refolded into its functional form, but not always. In general, remember that swings in pH, salt, and temperature can cause denaturation or unfolding. So a protein with its specific three-dimensional shape just unravels and it no longer works. It is biologically inactive unless it can be renatured, which is not always possible. As mentioned previously, The primary sequence, or the amino acid sequence, of a polypeptide will determine how a protein folds. However, it is very hard for scientists to predict exactly how a protein will fold from its primary structure alone. And it seems as though most proteins go through several states on their way to that stable three-dimensional form that is so vital to their function. So many proteins require help folding, and that help is provided by another group of proteins called chaperonins, or chaperone proteins. These are barrel-shaped proteins that assist in the proper folding of other proteins. And you can see a chaperonin illustrated on the left. The chaperonin protein has a cap that can come off, and that will reveal an inner compartment, much like a barrel, in which a denatured protein or an unfolded protein can enter. The cap can be replaced to provide this very isolated environment that is often essential for the proper folding of the protein. And then, once folded, the newly renatured protein in its functional form can be released from the chaperonin. There are some genetic disorders that are a result of chaperonins that are not working properly. For example, some forms of cystic fibrosis are due to mutations in chaperonin proteins. Cystic fibrosis affects the lungs and the digestive system and causes difficulty breathing and thick mucus to accumulate, decreased immune function, and frequent respiratory infections, all because a chloride ion channel is not folded properly or is not in the right position. There are many different mutations that can lead to cystic fibrosis. However, it appears that some forms of cystic fibrosis are in fact due to chaperonin proteins that are not functioning properly and are not folding the chloride ion channel proteins as they should be folded. It also appears that Alzheimer's, a neurodegenerative disease, is sometimes due to defective chaperonins. In Alzheimer's disease, the cells of the brain and spinal cord are lost. Many causes are unknown, but as Alzheimer's progresses, you have misfolded proteins that clump together and form plaques in the brain. If chaperonins were functioning properly, perhaps they could take care of those misfolded proteins and correct their form.